Welcome everyone to the EECS Colloquium. I'm Eric Paulson here on the faculty, and welcome you on behalf of myself and my co-chair Eli Ivanovich, who's right down in here in front. Let me try to give the time to our speaker today. This is very exciting. Many of you have maybe had a chance to encounter our speaker's work or meet her personally. You might have known her, maybe she was a PhD student at Stanford. You might have known her during her time as a postdoc researcher here in our very own CS department here at Berkeley. She says some of the furniture looks the same in this room, so I'm not sure about that. You might know her as a professor of computer science at Stanford University. You may have encountered her work in the more than 200 publications in a wide range of fields, science, cell, and nature genetics. You might know her as Time Magazine's 100 Most Influential People in 2012, as Newsweek's 10 Most Important People, as a member of the National Academy of Engineering, a fellow of the American Academy of Arts and Sciences, a recipient of a MacArthur Fellowship, the Genius Award, as co-founder of Coursera, who many of you have encountered, obviously the largest platform for massively open online courses used really all over the world. She's also been the chief computing officer at Calico. It's the alphabet company you may have encountered within the healthcare space. And I think what we'll be hearing about here, this is the really just fascinating career that she has had, and the impact is remarkable. And we're really, really honored to have her here today. And with her position, really, I think she's going to be talking about this latest role with the company where she's CEO and founded. In Citro, we're looking at machine learning techniques and how it can be applied and really further medicine in ways that I think has the possibility to change healthcare and just health and well-being for society radically. I will stall for a couple more seconds as she hits some buttons. But join me in welcoming Daphne Koller, Dr. Daphne Koller.
Thank you. Thank you very much, and it's a real pleasure to be back in this room, despite the furniture not having changed. So maybe as a little bit of a preamble and following on this very generous introduction, so thank you for that. I've spent the bulk of my career at the boundary of two disciplines, machine learning and biology. And I use the word boundary judiciously as opposed to the word intersection, because for most of my time in that space, there really was no intersection. It really was a boundary. You could do kind of data science light on biological data sets that were kind of small and bespoke, or you could do machine learning on various other data sets, but there really wasn't very much of an intersection. And one of the reasons I'm back to doing this and doing what I'm doing today is because I feel like now there actually is the opportunity to create a remarkable intersection of these two disciplines with tremendous opportunities. And I'll come back to that a little bit at the end of this talk about what it is that I think is part of the larger arc, if you will, in terms of those two disciplines. But let me start with what we're actually doing right now. And the focus of the work that we're doing right now is on drug discovery. And you can think of drug discovery as from the lens of the glass half full or the lens of the glass half empty. This, which worked a moment ago. Okay, okay. This is the glass half full. So in the last, century-ish or so, there has been tremendous amount in identifying new therapeutic interventions that have brought meaningful, hugely impactful benefits in health to people. And you go all the way back, it's the earliest vaccines, biologic therapeutics, which allowed us to move beyond small molecules into carefully engineered proteins that have a much better specificity and hence safety profile, and allowed us to target cancers and autoimmune diseases and a range of other things in a very fine-grained way. The most recent wave of cancer immunotherapies in which the immune system is unleashed to tackle cancer using the body's own defense mechanisms. Some of the recent work on on really leveraging our growing understanding of the human genome by identifying specific variations in the genome that can be targeted by therapies. And that happens in oncology, using a lot of the precision oncology work that's happened. But it also happens in other areas, other therapeutic areas. One of my favorites here is the remarkable work by Vertex in cystic fibrosis, whereby identifying 3 distinct genetic lesions in a single gene called CFTR with a different drug to each one of them, there's now basically a drug that provides an effectively normal life to 90% of cystic fibrosis patients, where up until that would have been a death sentence at the age of 20. So this is just an amazing set of accomplishments for this field. So that's the glass half full. The glass half empty is this one, which is, it's a law that's come to be known as Eroom's law. So all of us in this room, I'm sure, have grown up riding the tidal wave of Moore's law. You'll notice that Eroom is the inversion of that. And the reason it's the inversion is because this is on a logarithmic scale, the number of drugs approved per billion US dollars in R&D spend. And you will notice that that number has been decreasing exponentially year on year for the last 70 years, despite all of the advancements that I've talked about and many others, and nothing seems to be changing that trend line. And clearly this is not a sustainable model and is one of the major drivers for the for all the discussions that we're currently having about drug pricing and so on and so forth, is that fundamentally it is really expensive to make a successful drug. So how expensive is that? About 1970s, it used to cost maybe about $100 million to make a drug, a successful drug, and now it's over 2.5 billion. And in fact, I was talking to someone from a major pharma company who told me that since the number that we got in from this paper, which was a couple of years old, the number is now 3.2 billion. So here we are, exponential growth in action. And so what is it? It's not the case that a single drug that cost $100 million now costs $3 billion to make. People will tell you that clinical trials are now more expensive and patient recruitment is more expensive and you have to track them and FDA regulations and blah, blah, blah. And all those are true. That's really not the fundamental problem. The fundamental problem is that we just don't know where we're going. And we start out at the earlier preclinical phases with a whole slew of hypotheses about different genes and different pathways and different targets and so on, and then invest resources in a lot of them, and most of them don't pan out. And throughout the entire pipeline, there's tremendous attrition at every step along the way, so that at the end of the day, the one successful approved compound carries on its back the weight of all of those other drugs that never made it. And when you think about how much money and time you spend on going down wrong paths, if you could reduce somehow the number of wrong paths that you explore, then that potentially could bend that curve that we just saw. So if you think about this from the perspective of those of us who sit in this room, this is fundamentally a problem of prediction. If you had a little crystal ball, even if it was a murky one, that would tell you at the intersection of do I do this, 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 or this, that this one is more likely to work than those, you're not going to get it right every time, but maybe you can at least get rid of some of the really bad failures and avoid spending time on those. And machine learning, fortunately, has gotten really good over the last few years at making predictions. And so if you think about that, maybe this is a place where you can deploy machine learning to do things differently. So the driving philosophy behind what we do or what we're trying to do at In Sitro is that all of these decisions that are currently being made, by and large, are being made by a combination of historical heuristics, something called the Lipinski rule of 5 that says that a molecule that satisfies these 5 threshold criteria is a drug and other things are not a drug. And we know that's not true, but people continue to use it. Or gut heuristics about, oh, I believe that this is the pathway, amyloid beta is the pathway for Alzheimer's disease. And all of these are very qualitative and unvetted and unoptimizable. Whereas if we could create predictive models that we could optimize to achieve a higher level, to achieve a certain level of predictive accuracy, maybe that could help us choose a better path. So the goal of what we try to do is to identify areas where if you had a good predictive model, it would actually really help bend that cost curve. Second, machine learning, given the right data, is actually a good solution to the problem. Much as I'm a big fan of machine learning, it's not the silver bullet for everything, so you have to pick your problems. And the third one is that there are technologies the third one is based on the fundamental recognition that machine learning is only as good as the data that you give it. And in fact, the more powerful machine learning models that we employ today are even more susceptible to bad data than the simpler, more forgiving machine learning models of the past. And so found data that people say, oh, we're going to scrape data off with the supplementary materials here and there, or use the data that's publicly available, is very rarely good enough. And so what we also need is to have technologies that can generate the right data that's fit to purpose at high quality and at scale. And the other aspect of this, and I think this is really important, is to do this right, you need a very special type of organization where you break down the traditional silos between the life scientists and the computational scientists, and they don't, you're no longer in a world where the life scientists produce some data with some experiment, throw it over the wall to the machine learning people who do some stuff with it, send some results back. You really need to have people working as a single integrated team. And so really trying to build a company that from a cultural perspective is very, very different to anything that's been built before. So that's a very broad mission statement. And the rest of the talk, by and large, will be much more concrete and focus on a very specific problem that we're tackling first, which is the one that if you look at a sensitivity diagram of what could most move the needle on pharma R&D, it is this problem, which is the problem that most drugs fail in phase 2. And they fail in phase 2 because of efficacy, and that is because the drug is going after a target that does not modulate the disease. And so the question that you're basically answering incorrectly in most cases in clinical trials today is, what will this intervention do when I put it in a human? And if I'm able to make that prediction before I undertake the very expensive cost of a clinical trial, that's a very big cost saving right there. So right now, and this is a bit of a caricature, but not much, this question is mostly answered via the workhorse of biological discovery, which is a model system, which is oftentimes a mouse. And not only is it a mouse, it is often what's called an isogenic mouse, which means all of the mice are exactly identical, and furthermore, they're homozygous, which means both of their chromosomes are identical. It's a very funky mouse. I mean, a lot of these mice just die at an early age because of lymphoma or leukemias because of the fact that they're homozygous and they don't have enough, their chromosomes are so weird because of that. So this is what's currently used, and it turns out that mice are, in addition to these specific mice being funky, mice are just not a very good model for most human disease. Mice do not get atherosclerosis, they do not get Alzheimer's disease, they do not get schizophrenia or depression, or all of these are human diseases that don't exist in a mouse. Glaucoma doesn't exist in us. Mice don't get most human diseases. So in order to generate disease models that are mice, you really often have to stretch the bounds of credibility. So I'm going to give you two examples, both from the neuroscience space, just because that one's easy to pick on, because the models are just so bad. The mouse models for Alzheimer's disease is a model where they take the defective human protein that doesn't even exist in this form in this mouse, called amyloid beta, and you put it in toxic quantities into a mouse brain. And it is toxic, so the cells begin to die, and then you have a drug that basically clears the amyloid beta from the mouse brain, and then the mouse gets better because you no longer have the toxin. That's great, but it's a model of amyloid beta toxicity. It's not a model of Alzheimer's disease. A second one, which I think is even funnier or sadder, is the model of major depression, which is called the forced swim test. You take the mouse and you put it in a bucket, and you force it to swim until it gives up and drowns. And the premise is, if it swims longer, it's less depressed. Yeah. So, which kind of explains why there have been so few drugs that have been created for the neuroscience space in the last few years. So, that we're not the first to make this observation, and really what you want is to use humans as a model for humans. And since you don't get to experiment in humans in most cases until you get to the end stage clinical trials, what the field has come to do is to think about experiments of nature. So each of us is an experiment of nature, where nature has perturbed many of the genes in our genome and tweaked them in one direction or another. And we can look at the genotype, we can look at the phenotype, the traits of those humans, and ask, if I tweak this gene, does it make people less susceptible to the disease, have a slower progression of the disease, whatever? And that is an experiment of nature that one can then use as a way of guiding drug targets that are likely to be more effective. And sure enough, if you look at this paper, and this is one of a series that have repeatedly done this analysis over the years, and you look at drugs that target a gene that has a genetic association with the disease in question, you are twice as likely to have a successful drug come out of that than for drugs that do not target a gene with a causal connection in human genetics. So 2x for an industry whose overall success rate is on the order of 5% is a huge deal. And so this is a really important result on how we select drugs by using human data as a way of guiding the selection. So that's great, and in fact, this is a place where the technologies that have emerged over the last few years have really created an incredible opportunity for us to get a lot of data that is relevant to exactly this problem. So this is a graph, this is a Moore's law graph, not an Eroom's law graph, because you'll notice it's on a logarithmic scale, but it's up and to the right. And it's the number of human genome sequenced since the very first human genome in 2001. And you can see that not only is this growing exponentially, it's growing exponentially twice as fast as Moore's law. So depending on whether you believe that this trend will slow down to be only as fast as Moore's law or will continue with the current historical trend line, the number of human genome sequence that we'll have in about a decade will be on the order of 100 million to 2 billion, and that's a lot of data. Now, genotypes on their own are useful because you can look, for instance, at variations that are depleted in the population, say those are probably bad for you. But obviously, it's much more useful if you combine it with phenotypic data. And this is an area where there's been also a fair bit of growth, although not quite as much as on the genetic side, in resources like the UK Biobank, for instance. So this is 500,000 UK volunteers who went in for a full 2-day physical exam where they did full medical histories and various functional tests like cognition and hearing and eyesight and walking and blood biomarkers and urine biomarkers. And for 100,000 of them, they even have full body imaging and brain imaging and fMRIs. It's an incredible resource. And this is in the UK, which was one of the first, and I still think one of the most successful, given rise to thousands of publications understanding the genetics of different traits. There's now data sets that are emerging along those lines in multiple other countries, including here in the United States. There's a data set called All of Us, which is supposed to have 1 million people in the cohort, and there's many others that are coming up in other countries as well as in other just ecosystems. So this is great. And you can take the phenotype data and the genotype data and do now what is called association, where you look at the extent to which a genetic variation in the genome aligns with a particular change in the phenotypic trait, and that generally, by and large, suggests causality. Now, we can quibble about the cases where the causality is not quite so clear, but by and large, as a first cut approximation, since the genetics precedes the traits, if you see a change in genetics that associates with a change in the trait, it's the genetics driving the trait. So we can talk about exceptions, but it's a pretty good approximation. So this is great. And you would think that we are done, but we're not, because the truth is that while we have a lot of these, so we've had a lot of these associations in the last decades since we started to do these genome-wide associations. And people have found genetic causal variants to a very large number of very diverse traits. Some of those are diseases like multiple sclerosis or Alzheimer's. Others are quantitative, more biomarker type traits like triglycerides or LDL cholesterol or QC interval in your cardiac rhythm, QT, interval. And some of them are even not diseases. diseases, like educational attainment turns out to have a pretty solid genetic basis. So this is great, and we have a ton of these, and you would think, okay, great, each of those is now a drug target, and we can now go after the genetics of multiple sclerosis or Alzheimer's, and we're done. Except life is not that simple, because each of those genetic variants that have been tied to the phenotype is complicated. Most of those are just polymorphisms that tag an entire region in the genome. That region can contain 3 genes, but it can also contain 30 genes. And you don't even know how big the region is, because sometimes there is the variation in one area that actually is related to actually acts on a gene that's very distal, that's like hundreds of thousands of bases away, and you don't know. And many of these variants are unclear. They don't sit inside the gene, they sit adjacent to genes, and their effect sizes are very small. So when you look at the population aggregate, a typical effect size can be plus or minus 4% in the trait, which is not very significant if that's really the entirety of the effect. Now, the fact of the matter is that if you hit the gene over the head with a hammer, like with a drug, the effect sizes can be really large, but you don't know that by looking at the strength of the genetic association. So it makes it really hard to look at one of these loci, and there's usually dozens or hundreds of loci for each trait. Each of those has, multiple genes associated with it. Effect sizes are very small. And say, okay, I'm going to start a drug program on that one, when you think about the fact that each of those drug programs is on the order of a couple $2 billion. So you can't just go out on a limb and just try them all one after the other. So what do you do? So, and in fact, it turns out that this is a serious issue. So even with all of our ability to try and pick these genes, if you look at the set of targets that the graph that I showed before, which is this 2x improvement in odds ratio for genes that have genetic validation, if you separate this out into 2 categories of genes, what's called Mendelian genes, where there is a clear, strong one-to-one association between the gene and the trait, versus the ones that come from these what's called polygenic traits, where you have many, many genes that are associated with the disease, you can see that the numbers get very different. The odds ratio for the Mendelian is more like between 2.5 to 3, and the odds ratio for the stuff that comes out of the genome-wide association is below 1.5. So there's still signal there, but it's much weaker and much harder to tease out. So we still have the question of how can we better leverage our understanding of human genetics to find better drug targets. So this is a place where we're going to bring in a very complementary orthogonal set of tools that goes beyond human genetics to the ability to create very high content biological data at the cellular level that hopefully brings us much closer to the causal biology of how the variant actually acts on the disease, and that might help us pick variants that are actually much more relevant. So let me give an example of that just to make this concrete. There's many more that I could have given here, but this one is just really beautiful and very visual. There is a region on chromosome 16 that for whatever reason is subject to copy number alterations in a relatively substantial number of humans. And in some humans it's deleted, and in other humans it's duplicated. And when it's deleted, it has a 75% penetrance for autism, which means if you have that aberration, you have a 75% chance of getting autism. And if you have the duplication, you have a 40% chance of getting schizophrenia. Now, that region contains 25 genes and sundry other genomic elements that might also be functional. No one knows which gene in the region or genes drive one or more of those phenotypes. So this is where we kind of were until a couple years ago, at which point a group with support from the Simons Foundation took 3, this is one of those miracles of science. At this point, we can take either epithelial cells, skin cells from any one of us here, or white blood cells, and reprogram them into what are called stem cells. And stem cells are these pluripotent cells that exist at the earliest stage of development and can then differentiate into any other cell in the body. They can differentiate into neurons, they can differentiate into cardiomyocytes, they can differentiate into hepatocytes, all sorts of different cells. And we can do that with any one of our cells here so that we have a cell that is a neuron, but that has the genetics of a specific individual human being. So this gives us a tool to understand how genetic variation of a specific human affects the cellular phenotype. So they took 3 of those IPS lines, they're called induced pluripotent stem cells, from the deletion patients and 3 from the duplication patients and 6 or 7 healthy controls, and reprogrammed them to get the iPS cells and differentiated them into neurons, and this is what you see. So even with the naked eye, you can see that the pictures on the left, which are the green, are the neuron stained, so using green fluorescent protein, you can see that there's significant excess of synapses and increase in axon length. And on the right side, which is the duplication, you see a significant depletion of synaptic arborization and axon length. And so this was like an experiment done by hand, 3 cell lines, and so they had some really, really sad postdocs sitting there counting and measuring axon length and so on. And even with the sort of manual quantitation, you can get to fairly significant statistical differences between those measures in these 3 individuals on each side. So this is really, I think, an incredible, oh, and I forgot to say that, that this aligns with what we see in postmortem brain samples from patients with these genetic variations, because you see more synapses in the deletion patients and fewer in the autism in the duplication patients. So this is really an amazing feat in scientific understanding, but by itself, it doesn't tell us how to then build a drug. I mean, we now understand something about the process, but fine, how do you turn that into a drug? But imagine if we could do this not for 3 cells for each type or for 3 individuals, but we could do this for 100 of each type. And I should tell you, this is not a rare syndrome. 1% of autism patients are 16p11 and 10% of schizophrenia patients are 16p11 too. So now you can do this for hundreds and in fact use machine learning to figure out what it is that makes them different from each other in an automated way, and then use that as a way for now screening for interventions that make the cells on the left or the cells on the right look more like the cells in the middle. Now, is that guaranteed to work? No. But would I be confident in an intervention that did that enough to take it forward into clinical development? Probably yes. So that's how you could potentially use this type of analysis to identify new drug targets or new drugs. So what we're really leveraging here is really 3 revolutions that have all come about in the last 5 to 10 years. The first is the one I already mentioned, which is the ability to take iPS cells from regular people with or without disease, with different genetic backgrounds, and create normal cells. So these are not the cancer cells that one typically works with, whose relevance to pretty much any human biology is tenuous at best, because even if they were kind of like cancer at the beginning, by the time you passage them many times with the genomic instability you have in cancer, they begin to have 8 copies of one chromosome and 0 copies of another, and they're just like, they're not even human anymore. Here you can actually use cells that resemble real human cells with the genetics of real humans. You can perturb them in very fine-grained ways using technologies like CRISPR that allow you to genome edit those cells and turn them into something that is different, and thereby increase the disease risk or potentially decrease the disease risk and see what happens. And then finally, you can phenotype them using these beautiful images that I showed you before that allow you to really measure many, many, many aspects of the cells, including ones that you wouldn't necessarily have anticipated up front are the most relevant to the disease. And because of the ability to use automation and microfluidics, you can actually do this at remarkable scale. So you can generate tremendous amounts of data that will really feed machine learning algorithms of the new type. And so you end up with millions of images that look like this, and they're really beautiful. But your eyes glaze over when you look at these, and it's very difficult for a person.
What are the axes?
the axes.
And in other words, is this some abstract?
No, these are neurons.
These are just neurons.
These are neurons and
Okay.
synapses, and I think the synapses are the axons are the green. Honestly, I forget what the red and the blue are. But this is just a fluorescently tagged microscopy image. And I forget what we tagged with each color, I'm sorry.
What's the interpretation of the colors?
So each of them tags a different type of protein in the cell and therefore marks a particular cellular compartment. So I'm pretty sure the greens are axons. I think the blues are the nuclei, I believe, and I think the red are synapses, but I'm not 100% sure.
Can you image neuroreceptors like dopamine also?
I don't know the answer off the top of my head. It depends on whether you have an antibody that is specific enough to tag dopamine receptors and not other things. And the state of the art on antibodies changes and they come up with better ones. But if you have an antibody, it's very easy to tag it with fluorescent and measure this.
You all agree that, if you take a machine, you take a neural net, and you start to show somebody thinking about something. This is the pattern in the head, and something where somebody else and then he's thinking about something else. This is pattern in the head. Machine learning will be able to then seeing a pattern in your head, what you are thinking. That's
from functional MRI. This is not
no
this.
No doubt. But you in some way sound like machine learning. How will it capture the causality? In doing what is the disease, there is a lot of causality.
Okay, let me hold that question. We'll get there. It's a good question. Right now, we're simply associating. Hold that question, and I promise we'll answer it. And if I don't, then you can ask me again. Okay. So, how do you interpret millions of images that look like that? So this is where we have a brief review of machine learning, and I know this is familiar territory for a lot of you, but bear with me while I go through it. It's like a 3-slide sequence, and it has a point to go through it. So this is the progress in machine learning, which actually was contemporaneous with the progress in some of the biological tools that I mentioned, also in the last 8 to 10 years. And I actually did a lot of computer vision when I was still at Stanford. And when I started to do computer vision around 2005, and I could tell you that we would show this image to a computer and ask it, is there a bear in this image? And it would say, I don't know. So fast forward 7 years to 2012, this is the era of ImageNet, where by a massive community effort led by my colleague at Stanford, Fei-Fei Li, we had 1,000 classes with a few hundreds or thousands of images of each class. So that was enough fodder to actually train reasonable machine learning algorithms. And so you would show the computer this image and it would say, yeah, maybe there's a wolf. Now there's no actual wolf in this image, but the bear is kind of wolfy looking, and at least you're in the same general ballpark. So fast forward 2 years to 2014, and the actual caption for this image in a state-of-the-art algorithm was a brown bear swimming in the water. You can probably see that the bear isn't actually in the water, it's just close to the water, but it's a pretty darn good caption. And in 2017, 3 years after that, 2 brown bears sitting on top of rocks, which is a completely accurate description. Today, 2 years after that, there's quantifiably, repeatedly, a computer can caption images like this at performance that transcends that of a human. And despite the fact that this is a task that we as humans have been trained to do since birth. So when you think about the problem of looking at the cell images from the previous slide, where none of us have been trained to do that since birth, or even more so the problem of looking at RNA sequencing data from those cells, which no one can really understand by looking at them, the ability of a machine to make sense from the type of data that we're collecting is almost certainly going to transcend that of a human very, very quickly. So how does modern day machine learning work? And again, I know this is a repeat for a lot of this audience, but again, there's a point to this. So what's made the machine learning improvement in the last few years move as quickly as it did? In the olden days, by which I mean 5 years ago, we used to have poor downtrodden grad students who used to spend large chunks of their time doing what's called feature engineering. And feature engineering is when people would say, okay, I need to look at edges that look like this and textures that look like that and little circular things that look like noses, and then the machine learning would kind of put them all together using some fairly straightforward combination like logistic regression and would make the prediction. Turns out that people are actually really bad, even when they can perform a task, at telling the computer how to perform that task. And specifically, they're really bad at defining salient features. Today, we give the raw input to the computer, and it trains itself to build more and more elaborate and complex hierarchies of features, so that at the end, you end up with a very compressed feature representation that is much more predictive than anything a person could have come up with, and can make very subtle distinctions like between an Arctic fox and an Eskimo dog, where a person would be really hard-pressed to define how you would possibly distinguish between those two. So that's one aspect of this. The other aspect, and the reason why I really bothered to show this, is that the other thing that occurs as a byproduct of this is that if you start out with images that by and large sit in a very high dimensional space, so let's for simplicity assume that it's a 1,000 by 1,000 grayscale image, so the image sits in a 1,000,000 dimensional space. At the end of the day, that last layer of the neural network is, let's say, 100 dimensions, 100 numbers at the end. So what you've done is you've taken a 1,000,000 dimensional vector and you've reduced it into a 100 dimensional vector. So from a geometric perspective, what that means is that you've taken and defined a 100 dimensional manifold in a 1,000,000 dimensional space. Now, I can't draw that, but I can draw a 2-dimensional manifold in a 3-dimensional space, so you can think of it as this way. And the important note is that there is an infinity, a continuum of 100-dimensional manifolds in a 1,000,000 dimensional space. And they have very different, they look very different to each other. By training the predictive model to make certain types of distinctions, like between a fox and a dog, or between a car and a dog, you end up creating an architecture in that low-dimensional manifold that is required to put two trucks next to each other in that low-dimensional space so that it can make the same prediction for both of them. Even though at the pixel level, look at them, they are completely different. Different color, different pose, different texture. There's really no similarity if you think of a pixel-to-pixel mapping. And because the feature hierarchies are shared, it's actually going to also discover relationships between classes that are not the same class. So it's going to put the tractors and the cars next to the trucks, even though they're not the same, because they have wheels in common and things like that. Whereas dogs and cats and turtles are going to sit in a very different region of the space. So it discovers some kind of semantic distance function in this low-dimensional manifold that is not obvious in the 1,000,000 dimensional space. So why is that important, and why did I bother going through all this? Because this is really the fundamental thing that we're going to be leveraging. So think about the ability to, say, collect IPS cells from the 100 or so people that are in this room, for instance, and imagine that we had a spectrum of disease burden on this. And so we have 100 IPS cells, IPS cells from 100 people, and they're sitting in this very, very, very high dimensional space of images of those cells. And it's very difficult from such a sparse sampling to make any sense of the architecture of the space. But that's where we get to use tools like CRISPR engineering to considerably expand the diversity of the genetics of those cells by introducing additional mutations, for instance, that are more likely to, say, cause the disease. And so all of a sudden, you have a couple hundred points in the space. You might have hundreds of thousands of points in the space. That we can now use as input to the machine learning to discover a manifold that captures what we think of as the cellular state. Now, that manifold is now something that we can now used to understand the architecture of the original set of humans that we had to begin with. And specifically, distance functions now make a lot more sense than they did in the original million-dimensional space. And so clusters emerge that don't really emerge if you think about the original set of cells. And those clusters correspond to groupings of patients that may have been very similar at the clinical manifestation, but are very different at how they appear at the molecular level. Now, I think there's an important analogy that we should keep in mind, which is precision oncology, where up until not that long ago, we used to think of breast cancer as one thing, until we actually were able to collect around 2000-ish enough molecular data from enough patients to recognize that clusters of breast cancer, breast cancer patients are very inhomogeneous in how they manifest at the molecular level in their tumors, and that there's subtypes of breast cancer that are much more similar to ovarian or pancreatic cancer than they are to other types of breast cancer. And specifically, that's allowed us to move from a model where we treat multiple very distinct diseases that might just manifest similarly in a clinical way using now different drugs as opposed to one drug. And I have colleagues who were at Genentech at the time that they created Herceptin, which is one of the earliest precision oncology and one of the still most transformative precision oncology drugs. And they will tell you that if you'd given Herceptin to the all-comers breast cancer population rather than to just the HER2 positive breast cancer population, you would have needed 8,000 to 10,000 patients to discover any shred of statistical significance, and the clinical trials would never have worked. So by segmenting the patients in this way, we're able to figure out what those subtypes are, which we can't do today because people don't like, for instance, to let you take biopsies of their brain. So we don't get molecular data for multiple patients in some of these other diseases. And by segmenting those patients, we can now look for interventions that revert the cells from an unhealthy to the healthy state. So to your question about causality, the causality comes in when I put the intervention into these cells, because those are intervenable. It's a cell-based system in which I could actually perturb the system, and ask, if I make this intervention, does it revert the yellow state, the cells in the yellow state, to the cells in the green state? And assuming that we believe that that correlation between the green state and the healthy human and the yellow state and the unhealthy human is a meaningful one, like for instance in the synaptic arborization, if that intervention actually has that effect, then that's something that we might believe. Okay. So that's sort of the big picture vision of this, and we are only about a year and a bit old as a company, so a lot of work is still going on, but we have over 100 IPS lines right now. We have the ability to differentiate them into multiple lineages. We have different CRISPR modifications that we can take, and we have the ability to phenotype them in a variety of different ways. We also have extensive automation that allows you to do this in a way that is much more scalable than you can ever have by people pipetting, and furthermore with much higher quality than you get with people pipetting, because most of the variability that you get is in fact by human variability of how the experiment was performed. And so instead of telling you about these things here, I will tell you about them in the next slide, where you could actually see a video of the automation at work. Can we turn down the volume, please? Okay, thank you. So samples are retrieved. These are IPS cells. They are prepped for cell culturing. This is a liquid handling system. I know it's not R2D2, but it's still pretty cool. You put this in the cell line management system, which can actually culture hundreds of lines at once, and this is what IPS looks like when they're differentiating into different lineages. I only wish it was this fast.
IPS lines from patients?
Some of them are for patients, yes.
With neurodegenerative disease?
We don't talk about which patients. Then you put them into this wonderful piece of equipment called the LabSite Echo, which allows you to manipulate these cells without actually having a pipette ever touch them with, and since pipette tips are reactive, it's really important because it allows you to do an entire experiment in a single well, longitudinal experiment. And then you basically can take those samples, and I'm going to go back because I didn't get a chance to, sorry, this is like the coolest thing. This is acoustic sound waves that are manipulating the droplets and pushing them out of the media, and then they get held on the top using surface tension. You turn the plate around and you can do the experiment, and the cells continue to grow, which is really cool. And then, like I said, you take that and you put this into the microscope or the sequencer, depending on what assay you're doing, and you get really, really beautiful images like the ones that I talked about before. And what's also important is that this gives you not only scale, it gives you incredibly rapid turnaround because you have all of the pieces in place every time you want to run an experiment, and allows you to also capture extensive metadata because the entire lab is full of Internet of Things. So we capture everything, including temperature, humidity, the concentration of particulates in the air, the batch of reagents, the technician on site, whether the cells were on the bottom or the top of the stack of plates, whether they were on the edge or on the middle of the plate. All of that is captured and kept track of as the cells continue on their journey, so you can test for all of those confounders when you're doing the machine learning. So, so that's that. And then, of course, since we are half biology, half data, there's all the data side of this, which is all the data needs to be collected, curated, stored in the cloud, processed using state-of-the-art pipelines. There's a lot of data. It's basically, I think in the last 2 weeks, we have 20 terabytes beyond the 75 that we didn't just update the slide. So it's just massively growing. And there's some really cool machine learning pipelines that are not fun to write, but are incredibly important when you're manipulating images that are 20,000 by 80,000. And then just to show you that we can actually do machine learning on these, this is a toy machine learning example, but it's kind of cute. So these are HepG2 cells. These are a type of hepatocytes. And they were subjected to different types of chemical compound treatments. And the question is, can you look at the cells and know what was done to them? So infer the perturbation from the image. And this is more of like a showcase study for machine learning. It's not something you would actually use. But the point is, it took 3 months of work to get there because there was a lot of work that needed to be done because we're pushing everything to the boundary of what's feasible in terms of the machine learning pipelines and the microscope. But on the left, you see this is the state-of-the-art performance from the Broad's pipelines, cell profiler with a neural network on top of it. This is the end-to-end model that we trained, so you see the significant change in performance. And then the really cool thing is that we did that for chemicals, where, by the way, we're able to detect changes that a trained cell biologist cannot train. So the machine learning is outperforming the human. And then we did the same thing for a much harder problem, which is using CRISPR, but not just CRISPR, but CRISPR interference to modify the expression of 10 different genes in those cells. And CRISPR interference doesn't knock the gene out, it just knocks down its activity by 20 to 40%. So it's a very subtle change in the cellular state, and yet you can see that there is a pretty reasonable clustering of the cells, and now the performance is not quite as high, but now at least we have room for improving the models because we actually saturated performance on the compounds. So that's the next step of what we're doing. So this is kind of almost the end. I'm going to end in just a couple of philosophical notes. One is, I think, beyond any specific technology that we're putting in place here, whether it's iPS cells on the one hand or your extra special GANs or whatever on the other, what's really important here is the environment that allows life scientists and computational scientists to work together as a single integrated team, where it's not siloed and it's not one kind of subordinate to the other, but everything from what problem do we tackle, what experiment do we design, how do we build the assay, how do we build a machine learning model, and how do we derive insights is all done as a single team. And you're able to use machine learning to design experiments, help optimize the experiments, interpret the experiments, and the whole thing is a closed feedback loop. And I think that's really a critical part of what we're trying to build. The last piece of it is a little bit of a philosophical ending note, which is why I'm back in the space after having done Coursera and all those other things. If you think about, you look back at the history of science, and you'll notice that at different times in this history, different eras, there's times where a particular discipline, because of a new insight or a new technology, suddenly took off and a tremendous amount of progress was made in a very short amount of time. So in the 1870s, that discipline was chemistry, where we moved away from alchemy and trying to turn lead into gold and more into the periodic table. In the 1900s, it was physics, with the understanding of the connection between energy and matter and between space and time. In the 1950s, it was digital computing, with the ability to take silicone and get it to simulate the kinds of calculations that only biological brains had been able to do up until that point, and more. And then in the 1990s or so, there was an interesting bifurcation where there were two such disciplines, both of which suddenly took off. There was the field of what it would call data, which is related to computing, but not quite the same, because it also draws in elements from statistics and optimization and various other disciplines that came to produce what we call data science or machine learning or whatever. And the other one was the field of biology, or quantified biology, which is the ability to measure biology in whole new ways at an unprecedented scale. And that includes approaches like short-read sequencing and super-resolution microscopy and multiple other ways in which we can measure various aspects of biological systems. And they kind of evolved in parallel with not a lot of interaction between them, coming back to my earlier point about being at the boundary rather than the intersection of two fields. But I think the next era is something you might call biological data science something, or technology. I'm hoping someone comes up with a really cool name, which is really the integration of these fields, these last two fields, into one, in which we will be able to measure biology at unprecedented scale and in whole new ways, use the kinds of data tools that we're developing to, we as a community, I mean, to interpret what we're measuring, and then use that to now engineer biological systems to do things that otherwise they wouldn't normally do. And there's implications of this in many industries. In materials engineering, I think there's a tremendous ability to use biology to create new materials much more cost effectively and with a much richer repertoire than traditional petroleum-based materials. I think there's tremendous applications in agriculture, in making more sustainable food sources. We're already starting to see some of that in the environment and things like biofuels, or maybe a little futuristically, bacteria that actually manage to sort of sequester carbon dioxide, who knows? And then applications in human health, which is where we are. And I think there's just a tremendous wealth of opportunities here, and I think this is going to be the discipline of the 2020s, and I think there's just a lot of exciting things that can be done. Thank you.
Okay, I don't know if this is on. Okay, great, thank you. So many things to think about. Questions from the audience? Oh my goodness, it's going to go on. Okay, I'll try to get to everyone. Here we go. Thank you.
This is so
Oh yeah, you
interesting.
kind of look at, yeah, just.
Thank you for your talk. It's very fascinating, and the technologies in your company look incredible. I have two quick questions. One is that we know that disease and humans change over time. Are you using some of the machines in your company to collect time series data to develop dynamic predictive models? And the second question is, these machines look wonderful, and they seem very expensive. is the NIH paying like for it, or is it like pharmaceutical companies? And
Okay.
how did you make that happen?
Okay, so to the first question, there is actually two time series that you might think about. There is a time series at the cellular level and time series at the human organismal level. Time series at the cellular level, the answer is absolutely yes, and we're definitely developing both measurement assays as well as machine learning models that look at cells as they move and function, because you learn a lot more from that than just by looking at fixed cell samples. Longitudinal data on humans is, long-term follow-up data is generally relatively hard to get because people, even when they're participating in clinical trial, it's oftentimes of short duration and only begins after you're already sick. So that's where you get into some of the questions regarding, can you go back and look at electronic health records longitudinally and really try and analyze disease progression. And there are so many issues and artifacts with electronic health records that honestly we haven't really looked at that very much. I mean, we do look at certain really lightweight aspects, like, you know, things like whatever, disease onset age and things like that. But really looking at time series data is we might get there, but today we're not there yet. As to your second question, we're not funded by the NIH. We're a traditional startup, which means that we're funded by investors. And yes, the equipment is expensive, but honestly, it's not as expensive as you might think. I mean, say the microscope is the sort of top-end microscope, and Opera Phoenix, it costs approximately $850,000, which is not insignificant, but it's also not the, oh my God, it's gonna, you know, it's not the nuclear accelerator type thing. So if you're careful about this and you know that you're making an investment for the future and you're gonna be using this thing as a workhorse, when you compare that to the cost of the $2.5 billion on a per approved drug, it's like a rounding error. So you're making an investment up front with hopefully something that will increase your probability of success from 5% to 6% you've paid for it.
Thank you.
Thank you again for the talk. When you guys are making genetic variations, is it mostly with CRISPR-I? Are you reducing expression? And second, is it random, or are you guys choosing specific gene targets and changing, either knocking them out or changing expression?
Yeah, so there is a proliferation of different CRISPRs that are emerging. There was a beautiful paper from David Liu's lab just like 2 weeks ago, which talks about actually substituting in at relatively high efficiency an entire region into the DNA. It's just a lovely tool. So what we use changes from you know, over time as new tools that are robust enough to put into this kind of scalable production become available. The question of do we do this randomly or do we do this in a more systematic way is an experimental design question, and there are arguments to be had on both sides. And that's something that honestly we're exploring and may end up being different in the context of different diseases, depending on how much you know about the genetics of the disease. I mean, if you know that this is a mutation, then you probably should go make that mutation.
Thank you for a great talk. This question goes to the business model. Are you planning to develop your own line of molecules, or are you taking third-party molecules and then applying the simulation and genetic information you've created in your machine learning process to develop more efficacy in the trial process?
So we're not so let me try and answer that in a slightly different way. We're not a service company to pharma in the sense that we're taking someone else's clinical trial and helping them decide whether to move forward with that or not. Whether you design your own molecules from scratch, use existing molecules maybe in a way that was different than the one that was originally tried out, or extract natural molecules from the environment is honestly bit of an orthogonal question to what we're doing right now, and also doesn't even begin to address the whole question of alternative therapeutic modalities that are not small molecules, like gene therapies and cell therapies and antisense oligos and peptides and so on. And so this is effectively a screening platform that allows you to find interventions and validate interventions in a way that will hopefully allow you to make prediction on its relevance to human disease. Which interventions we end up vetting will depend on the set of targets that emerge from this platform. So if you come in and say, look, here's a target, and there happens to be an amazing drug that targets this and is not being currently used for this indication, it would be foolish not to use that. But at the same time, there's nothing to prevent us from taking something that is a brand new target and designing a compound just to that.
So as someone who did a lot of pipetting at one point, I think this is really exciting, so thank you. I was sort of curious about, you mentioned how the mouse model is a pretty poor substitute for humans in a lot of respects, but iPSCs also have their own set of issues where they're not identical to human cells, and also you're cultivating them in a very non-human environment in a petri dish. So I'm sort of wondering, are there like a set of like areas or targets that you think are a particularly good match for this as over the mouse model? Because it seems like in some cases it might be worse.
No, you're absolutely right. There are clearly areas where a system level model, even if it's a mouse, are better than a cellular assay. The way I look at this is that there are clearly areas today where this is, the example that I gave is one where mice don't get autism, right? So I think there's clearly places where this is a much more appropriate model for human disease than the mice. And those are probably the ones we would go after first. And I mean, you don't have to be a genius to figure out some parameters around that. It would include things like the disease has a strong genetic basis. There's a relative one or a small number of cell types in which you can kind of cluster a lot of the disease. You have the ability to create those cell types in a lab environment by differentiation protocols. Some are much better than others today. So there's a list of such criteria. As I said, it's not rocket science to come up with them. What we believe, though, is that while that's a subset of the diseases today, this is a very rapidly moving field, and every couple weeks someone comes up with another even better disease model system for something. Some of them involve organoids, some of them involve organs on chip, some of them involve the ability to do prime editing as opposed to just knock out of a gene. There's multiple tools that are coming into play, and so in some sense we're riding a tidal wave of innovation that is generated by a large community of incredibly smart people that open doors for us every day. So things that are not accessible today might be accessible in 2 years or 5 years or 10 years. And so you just kind of opportunistically work on the things where this technology is good.
So part of your big data is collected by doing the CRISPR modifications.
Yep.
And then you can't really put those cells back into a human being.
No.
Can you?
No, of course not. I mean,
but
that's not the point.
So it means you have to.
I mean, you can, but that's not what we do. Not in this protocol,
right.
but yes.
So it evolves. So basically, you have to sort of guess based on what happens to that cell, what would have happened in a human being.
True.
And so how's that done?
That's where the machine learning looks at and tries to connect what we see in the dish to what we see in the humans from whom we have data.
Going to the back.
Thank you. This is a wonderful presentation. I have a couple of questions, please. One is there is an area in toxicology which is called risk assessment. One of the conundrums in this particular area has to do with how mixtures are assessed. And I can assure you right now, nobody knows.
With how what? I'm sorry, I didn't hear.
How mixtures of different drugs or different compounds are assessed, which is the reality in the environment. You are essentially, you have to evaluate all those. We don't have any handle on that at all. So that ties into this neural networking system, where in your case, there are all these big data, and then of course the correlations that are being developed. My real question though is how do you integrate kinetic factors or equilibria systems, which is of course the homeostasis of the organism itself?
Okay, I'm not sure I understood the connection between the first part and the second part of the question. So maybe to start from the end, on the homeostasis part, we're very much looking to the biology itself to model the biology, in the sense of we're not trying to build an in silico model of a cell and trying to figure out computationally what homeostasis looks like. We have cells that come from human-derived tissue, and we can see what happens to them and to what extent they, the phenotypes might correlate with human clinical outcomes. And if that correlate has to do with a change in homeostasis or disequilibrium, if you will, in the cellular homeostasis, that will hopefully manifest in the assays that we're measuring, and we will be able to say, okay, these are the sick cells are the ones where the homeostasis got broken, and is there an intervention that returns it to what the healthy cells look like? And so that's basically where we're taking a very, I would say, agnostic and empirical perspective on this, as opposed to trying to come in with a knowledge of we understand what homeostasis looks like and how to restore it.
We have time for maybe another question or two. It's throwable. That's
Very close. Very good throw.
So thank you for the talk. So I have a question about basically there are two very popular approaches to get a lot of data right now. One is you do imaging, and the other one is you do single cell sequencing.
Yep.
What do you think about these two approaches, like in terms of neuron disease? Like, I feel like once you do imaging, at the end of the day, you can come back to the genes as well, right? So like, what do you think about integrating these two approaches in terms of understanding this, or which just gives more advantage?
Yeah, you know, it's something that we think about a lot, and I don't know that there's a single answer to this, in the sense that that some cellular states manifest better in sequencing and RNA sequencing data, and others might manifest better in cell morphology. It also depends what you're staining when you're looking at the cells. So are you imaging just the overall cell shape, or are you starting to stain for individual proteins like synapses or you know, or dopamine or whatever? It becomes a much more subtle assay at that point, and you can really start looking at cellular localization. So I think different ones will play a role in different scenarios. Now, of course, there's also some really significant experimental and computational challenges that make these very different to work with, and so those factors also come in. So, for instance, sequencing is by and large much more expensive for various reasons because of the way the market has worked, much more so than imaging. So imaging is much more scalable, much more cost effective, but also rather more challenging to deal with, because, for instance, if you're thinking about neurons, and neurons crawl all over each other if they're healthy, and so how do you start to measure quantitatively aspects of neurons, like how they, you know, their axon length and so on? It's kind of hard to measure if the neurons are sort of all mixed together. So the imaging becomes challenging at that point from a computational perspective. So there's not a one-size-fits-all, and one of the things that we keep looking for is also new ways of measuring biology. And again, fortunately, that's an area where there's been a ton of progress in the last few years, with single cell sequencing being one of those. And I think we're going to see a lot more of that coming on.
What would be your advice for somebody who's still a sophomore undergrad?
But you're lucky that you're this young.
I'm not that young, actually. I'm just a sophomore. What would be your advice for someone who's still a sophomore, in order for that person to be able to cope with the fast progress of machine learning, whether it's in computer vision or biology? What the background would you wish you had back in the days if you could choose?
Yeah, so when the background I wish I had didn't exist when I went to school. So But I would say it depends on what your career goals are. If you want to become a pure machine learning person, I would say the single most important thing you can work on is to develop your math skills, because it's the one thing that is hardest to make up for. And I see a lot of people out there who merged with, oh, I know how to do machine learning, and what it means is I know how to download code for a convolutional neural network and plug some data into it, and I don't really understand what the model does or what it means. So really get a sense for the underlying math, and also become a good enough programmer that you could actually start to modify those models. I think the core skills are really what's going to withstand the test of time, because the models just change every month. There's a new model, so you don't necessarily have to keep track with all of them, but you have to develop the skills that allow you to pick up what you need as the models become relevant to your goals. The other thing I would say is, regardless of which domain it is you choose to apply machine learning in, you need to develop, I think, a certain level of domain expertise. And in that respect, computer vision is misleading, because we're all domain experts in computer vision, because we all can recognize objects in some sense, right? And I think for that reason, a lot of people have kind of said, okay, well, we don't really need to understand the domain. If you do biology or many of the other domains that are much more less natural to people, you really need to understand your domain in order to come up with models that are not the, let's take a convolutional neural network, toss everything in, and miraculously the machine learns what it needs to learn. That just doesn't happen. And so that's the other thing, is if you want to learn, if you want to apply this to biology, go learn some biology.
All right, I found this so inspirational and so optimistic about so many directions. So thank you for inspiring us, and hopefully we will change the direction of things. I think she will be around briefly, but she will split very soon. So if you have it quickly, you could engage with her very quickly, and then she will have to leave. So thank you again. Thank you.